Welcome to Tracing Your Family Roots. My name is Arlene Sachs. This is Sally Ann Sachs. And today we'd like to welcome Greg Lemon. Greg Lemon is a geneticist who worked on the Human Genome Project. Welcome, Greg. We're delighted to have you here. We're going to talk about DNA, but I think our talk is going to be a little bit different focus than we have in the previous. Welcome. I'm glad to have you here.
I guess what I wanted to say to you was, before we talk about the Genome Project and DNA, I want to say that my question is, can any of this help the genealogist and family historian? If yes, how? If no, why not? Because we're certainly hearing all sorts of stuff about how we genealogists should all run off and have our DNA tested. So I hope you'll keep that in your mind and enlighten us on that. So,
okay. Well, thanks for having me. Maybe I can turn the question around a bit.
Okay.
And say that what genealogy can do for the medical sciences is do everything possible to collect medical histories. Not just for yourself, of course, but for your relatives, so that the wealth of medical history is brought to bear on what you or your loved ones may be predisposed to. Now, that's a different answer.
Yes, it is.
So you're saying that's much more, talking to people is more important than getting the testing done?
Absolutely. At this point, the testing's in a very, very preliminary stage. And we have learned many things through the Human Genome Project, but the result when you walk into a physician today is that the most important thing is your family history.
Okay, but you know, for those who've heard the word the Human Genome Project, but we really don't know exactly what that is.
Okay.
Could we have just a little potted history of that? Or
sure.
Description?
Sure. We, genetically speaking, have this blueprint. I think you have the map behind us, such that all of our cells carry DNA. And at the nuclear DNA level, which is the type of DNA we're talking about, we carry three types of DNA, basically. We have mitochondrial DNA that we inherit from our mothers. We have Y chromosomes that are inherited along with that third type, which is the so-called autosomal chromosomes. And we have altogether, therefore, 23 pairs of chromosomes, 22 of which, chromosomes 1 through 22, are autosomes, and then an X and a Y. The Human Genome Project was about sequencing fully those autosomes, those 23 pairs of chromosomes.
Sequencing is a verb I don't understand in this context.
Sequencing is determining the exact linear order of the chemical entities that make up DNA.
Okay.
In this case, called base pairs,
okay.
A, C, G, and T.
So you didn't use the X and Y at all? Or you
oh, no, they were very important. They weren't just all of the chromosomes, chromosome 1, chromosome 2, all the way through to chromosome 21, 22, and X and Y were fully sequenced.
Oh, okay. So now we
And so basically, as of the year 2000, approximately, the very first person was fully sequenced. In other words, we got 3 billion base pairs in order.
Then how do you
DNA.
Determine what means what?
We have to look for associations. In other words, we know now that 99.7% of those 3 billion ordered base pairs are the same between you and I.
That's what makes us human.
That's what makes us human.
Okay.
But there may be, at the single nucleotide level, so just look at one of those positions out of 3 billion, there may be 10 million differences between us.
All right.
Now, 10 million is a very small percentage of 3 billion, but that's what makes us different.
So that's the blue eyes and the brown eyes and that kind of
That's
thing.
right.
Okay.
And now what we look for are the associations that say this one nucleotide difference makes a person prone to something, and people who don't have that change are not prone to that disease or that trait.
But that doesn't mean they won't get it either.
No, it's a statistical kind of thing?
Strongly depends on the change.
Okay.
There are certainly some changes that strongly influence the outcome, medically speaking.
Okay.
Most of the changes that we're now discovering have very, very weak effects.
All right.
Classically, you may have heard about things called mutations, right? Mutations are usually very strong predictors. And so up until the year 2000, roughly, most people focused, before the Human Genome Project was fully sequenced, they focused on mutations. And an example of that is Tay Sachs.
Right,
Okay.
or Stanley Diamond's beta thalassemia trait.
Absolutely. In fact, many of the traits that you hear about originally, classically in medical history, in medical genetics, are based on these very powerful single mutations.
And those we knew of before we knew about DNA, because we knew about them from family history.
Absolutely.
Right.
But the good news is, those are very, very rare, generally.
Okay.
What's far more common are these relatively weak effects, these millions of changes that together may mean something very important for us, but individually, we can't look at that one change and say, oh, that means you will have an increased chance of heart disease or breast cancer.
So then DNA testing for the genealogist probably has been oversold, it sounds like.
Well, DNA testing for the genealogist is certainly being sold by for-profit companies.
Right.
You've certainly spoken on this show about Family Tree DNA, a highly successful company making millions of dollars in revenues annually.
Truly.
Excuse me, tens of millions. And that's fine, that's the American way. It's relying on advertising, certainly, to tell you something that you're going to learn. And another example is the National Genographic Project. My mother is someone who has taken such a test. It's about $100.
Yes, they're always about $100.
I think studies have shown that's the right price point.
Yes, exactly.
And my mother, for example, was born in Germany to German parents, and she found out after paying her $100 that she is of the U4 maternal mitochondrial DNA clade, subclade, which basically puts her ancestry as Northern European.
So you found out the
So
whole
what?
lot.
She already knew that.
That was obviously, absolutely.
But now
There was
she has a
nothing
piece of
learned.
paper to tell her.
She's $100 poorer,
Right.
and she's confirmed, more or less, what she certainly knew to begin with.
So my question to you would be, can you, knowing what you know about the Genome Project and genomes, can you think of any way in which any kind of DNA testing would be helpful to the genealogist or family historian?
Well, does the family historian care about medical predisposition?
Okay, so that would be one.
That's certainly one. Every day we learn a little bit more. We don't know enough yet to make a lot of these common variations meaningful to your physician
Okay.
or your genetic counselor in general. Now, if something's running in your family,
Right.
so you've done the medical, you've sleuthing necessary to look through your ancestry, and you see a particular trait that is being passed on and is medically important, then probably we can help get clues as to what in your DNA is predisposing you to it.
So you're sort of turning it around. You're saying that the family's going to help the DNA rather than the DNA.
Yes.
I have a question.
Family history is incredibly powerful,
Right.
because it not only takes DNA into account, it takes the behaviors and the environmental exposure that people have tended to expose themselves to over the years.
I have a question for you from my 18-year-old granddaughter, whose mother is left-handed and her father is left-handed and her one and only sibling is left-handed, and Emily is right-handed. And Emily hasn't liked that. She feels somehow like an outlier. And she learned in high school genetics, or whatever they taught in high school, that if your mother is left-handed and your father's left-handed, she was supposed to be left-handed. It's a good thing she looks like the family. Why isn't Emily left-handed? What was wrong with what they taught her?
Well, let's assume those are truly her parents.
They are, okay.
And one thing I'd also, just as a caution, say to a lot of genealogists is there's a surprising amount of non-paternity.
Oh, yes.
And I'm sure you've spoken about this on your show.
Bennett has a phrase, unrecorded paternity.
Unrecorded paternity. But let's rule that aside.
Okay,
Let's rule that out anyway.
right.
First of all, identical twins, for example,
Right.
are not truly identical.
What do
I
you
hate to break it to you.
What do you mean? I thought the egg splits or whatever happens, they're supposed to have the same DNA, aren't they?
Technically, there was a point when they were down to the one cell stage when they were identical.
Okay.
But change happens.
After that.
After the two embryos have started their independent development, first of all, there are small numbers of DNA changes that occur at all times throughout your life, in all of your cells.
Okay.
Secondly, there's been slight differences in their environmental exposure.
Yeah.
And there's also something called epigenetics, where basically the way in which your DNA is read out.
Read out?
Read out meaning used during your life.
Okay.
May have slight differences.
Oh.
So there are all sorts of differences that accumulate between identical twins.
Now you said
Not as many as between any two individuals in general.
You said the DNA could change throughout your life. Now,
Slightly.
Slightly. So, but you're only taking the cheek swab. I mean, is it going to change in your cheek and in your stomach and your toes? And I mean,
There are slight differences, but they're so slight that most people are ignoring them right now.
Okay, so tell me the sentence to tell Emily after this show. She is right-handed because?
There are plenty of identical twins
Okay.
who do not share every single
And
characteristic.
just because both of her parents are left-handed, there are other things that come into play in determining whether you will be left-handed or right-handed, aside from the fact that your mother and father are left-handed.
Absolutely.
Got it.
now
Okay.
I've got a
I mean,
question.
just add
One
one
more.
more thing. Obviously, in utero exposure is a well-known influence on how a child will turn out.
Oh, okay.
Just for example.
That's very interesting. Okay.
Okay, so that
Now, good. Now we can lay that one to rest.
there's a project that we've heard about where they're trying to get all the people who lost family in the Holocaust to put in their DNA, and they're going to try to match that to bones that they find. I don't see how that's possible. I mean, if it would be my mother's father. I mean, first of all, I'm not the male line all the way down. It's crossing, and they say, "Oh yeah, it'll be possible." Would it?
I am a strong proponent of the Shoah Project, actually. I think that it's a very interesting nonprofit effort to help living people, primarily.
How does it help?
It may connect people to families, basically, that they didn't know they were connected to, or that the connection had been broken over time. Right?
Well,
Anyway,
how?
the
You may
genealogy.
find relatives.
How? I need to hear this in steps, okay?
Okay. Certainly any two relatives, in fact, going back to two identical twins,
Okay.
they will share a tremendous number of their individual variations will be shared.
Okay.
And as you go expand from an identical twin to a sibling, a brother or sister, to a mother or father, you will share less, but you will still share far more
Right.
than you'll share
With unrelated.
with unrelated individuals. Projects like the Shoah Project rely on that degree of similarity to estimate how related are you to someone.
So it would be the whole genome, not just the Y or the mitochondrial.
I can't speak for their project.
Right, but that's what
in
you're
general,
well, that's how it would
the
work.
field of finding relatives, or for that matter, the forensic use of finding perpetrators,
Right.
is increasingly moving away from just using 5, 10, or 15 markers towards using the millions of variations that are actually present over these 3 billion nucleotides
Okay, then
of DNA.
here's my other question. I have used the DNA studies from Family Tree in one specific way. My maiden name is Amder, and for 30 years I've been collecting Amders everywhere, and I have these clusters of family trees, plus what I call the main trunk that goes back to the early 1700s. The question that arose was, all of these clusters that have the same fairly unusual last name, did we all have a common ancestor? Now, those of us who descend from the main trunk know how we relate back to the middle of the 1700s, which is as far back as we can go. The others are interested in knowing whether they might also be part of that trunk. It would help guide their research in certain ways. So we've gotten the swabs and studied, and some of the people do. We get a 35 marker study, I think it is. Some do. Others with the same name don't, think they should, are not happy at the thought of unreported paternity or whatever. What, if anything, have we demonstrated? Has this been valid, or in what way, what do we learn from
Yes.
this? Okay.
You've excluded certain groups as being along that line of lineage. That's your main value, really.
Okay, so we cannot claim, though, that we have included, that we can't say that that DNA has demonstrated that this one and this one and this one all share a common ancestor.
You might.
But we haven't shown that. Is that what you're saying?
Ultimately, you'll have a statistical argument that says we have a certain probability of having shared an ancestor.
I see. Okay.
But if you go
And
back
as it turns
far
out,
enough,
the probability is far stronger on the exclusion side than on the inclusion.
Okay.
Because you can rule things out much easier than you can rule them in.
Now, if for some reason all of these people had all of their genome tested,
Fully sequenced,
then theoretically, would we show it, or would it still be just a higher statistical probability?
it will always be a higher statistical probability.
Okay. But it would be, the statistic, we'd get closer to 99.999 kind of stuff?
Absolutely.
I see.
And in a court of law right now, for forensic use, that's certainly valid and used every day.
I see. So when they say DNA showed this, really they're talking statistically. They've accepted a certain level of cutoff.
Yes.
Yeah, well, they've always, even in the charts that we've looked at, say there's a 99% chance that it's in 200 years, or a 99% chance in 500.
Do we take in psychology, when you do your experiments, if you get 95%, everybody's very happy. But I don't know at what level they're happy in genetic testing.
And that's true for the medical predisposition world as well. When we say someone might have a BRCA1, a BRCA1 mutation.
What's that?
That's the breast cancer mutation that's well known to strongly increase your chances of developing breast cancer or ovarian cancer. It's particularly common in certain populations.
Right.
That is a statistical argument, really. There is a predisposition, and so you say you have upped the statistics or the odds, but it is not a genetic sentence.
Right.
It's not your fate.
And could,
with 100% certainty.
So are there other things that they might find eventually? Okay, you've got this one marker so far that you're using, right? So are there other markers that they will eventually look for, that if they have all of them, that there's a much higher chance, or that they'll get it?
We hope so.
Okay, but you don't know yet. That's what you're looking
So
for?
the project we've started, this project called SNPedia.
yes, what is a SNP? Yes.
A SNP is a single nucleotide polymorphism, which basically means one single change out of 3 billion.
Okay.
And we called it SNPedia because it's like Wikipedia. It's a community organized project that collects all the information that have been published, all the papers that have been published, showing an association, basically a statistical link, between a single change and a disease or a trait.
Now I have another genealogy question. There's a group in the Jewish genealogy world that calls itself the Worth group. They all have the same Y DNA. But recently, they have supposedly an unusual mutation, very unusual. And recently they've done this, we've been told that Family Tree DNA has done a SNP test on all of them, and they all somehow relate with that. Does that mean that they looked at a single one of these many millions?
So the state of the art right now, but it's changing almost daily, is that we know about, as I said, maybe there are 10 million locations where you might have a change.
Okay.
And any one of us has maybe, like for the people who have been fully sequenced, and there are a handful right now, there are about 3 million changes are found in the average person out of this 10 million range.
Okay.
Most of the tests right now test 500,000 to 1 million. So probably what the Worth group is doing is reporting back the results of how many changes were found in the 1 million that were checked. And there's a particular technology called a DNA chip that right now you can have tested.
For somebody like me who sort of zones out as we go into this stuff, the bottom line is, if they are still all identical with this SNP thing, has that increased the statistical likelihood of their having it?
They won't be identical.
Right.
They will certainly share more
Right.
across the 1 million positions checked.
Right.
If they're related, they'll share more of the changes than they would have if they weren't related.
So
the statistics did improve.
okay, so the likelihood that they had a common ancestor has gotten greater as a result of their having come back with this SNP test? Showing
Yes.
this? Okay, but they still haven't really demonstrated that they have a common ancestor.
No, because, for example, many groups that have existed in somewhat isolation geographically
Right.
share a lot of these variations simply through inbreeding.
Right.
Now, that means a group, they are related,
Right.
but they're related in part through genetics because of geography.
Right, sure, because of but you see, one of the interesting things about this same group, which I'm going to, is they all were Jewish from Eastern Europe, with one startling exception, which we had on the show, which was a man from Puerto Rico whose daughter, when she did his genealogy and his DNA, thought it would come back with some kind of African descent because he was very dark-skinned. And he, lo and behold, had the same, even this, as these Jewish men from Eastern Europe, which, of course, led everybody to then scratch their head and say, whoa, what happened? So now everybody's wondering whether they should go back and look for that ancestor that they all had in Spain before the Jews were kicked out of Spain. Because how else would you account for this outlier in Puerto Rico? Do you have any thoughts on this subject?
There are many, many such mysteries.
Okay.
And it will just take time to work them out.
Okay.
We don't have the Y chromosome DNA for most individuals on the planet right now.
Right.
Right?
Right.
Nor the mitochondrial DNA.
Right.
Let alone the full genome sequence.
Okay.
We don't really know how much variation there is across the planet.
Okay.
We have some ideas, we have good guesses, but until a lot more information comes in, we won't be able to definitively figure out exactly what all the relationships are.
So when Family Tree DNA says that they have they've gotten so much Y DNA from Ashkenazic Jewish men that they now effectively have covered the Ashkenazic Jewish world in terms of DNA, is that a valid kind of statement?
It is and it isn't.
All right.
It's certainly true for that group, but the moment they say, now what does that say about everyone else?
But if they never talk about everybody else, but only talk about that group.
But they basically can say these things are shared,
okay.
but they cannot say these things are unique.
Right.
Okay.
Okay. I have it.
So, but would it be logical that one person with a seemingly 100% different background suddenly has a J2 or a J1, or that someone, a white person in the United States suddenly is matching somebody from inner Africa? I mean, do you think that would be possible?
Yes.
It would,
Yes.
even though there's no,
Known.
no known.
Yes, because we're still learning so many
Okay.
things about markers like the J2 marker.
See, these fellows who are doing this study talk about themselves as
J2 clade, actually.
as migration anthropological geneticists, that they're interested in migrations. And can genetics be used, the DNA sequences be used for migration studies validly?
Well, isn't that what they?
Yes.
Okay.
Some of this blends a little into the discussion of race and ethnicity.
Okay.
And right from the outset, I have to say, most people will say, yes, you can use these markers to say you have some Indian ancestry or you don't, or so forth. But remember, race as a concept is a social concept.
Right.
It's not a scientific one.
Right, exactly.
So we wind up saying there are some things certain people will share. It's not because of something called race, it's because of their genetic changes and their family history and their environment and so forth.
Okay, sure. All right. Yeah, what happens though that becomes very interesting is when they've done these Ashkenazi migration studies, we have an exact parallel with a name study. What's it called? I've forgotten the word, it's the study of names, that Alexander Bader did, showing the development of Jewish names in Europe, and a cluster coming from this area that was once called Khazaria, and other places, and showing the migration by looking at where the names were, and it just matches like an overlay with the DNA.
It will match often.
It does.
And there's a website, in this case I forget the name, where you can type your surname.
Orthography, okay.
And I think it will map out your ancestors at least back 1,000 years.
Really?
Completely for free.
Well, it has to.
You have to remember that website
That's
for us.
interesting. I mean, for many of us, we know where our name came from.
Well, we didn't
There was
have
a town 100 years ago, or 150
Well, you know,
years
most
ago.
Jews didn't have family names until the authorities forced them on us at the beginning of the 19th century. So they're Johnny-come-lately things, and people didn't really have much fidelity to them most of the time.
We're really down to under a minute. Is there something that we've really missed that you want to bring out quickly while we still have the time?
In only a minute? The Human Genome Project is a fascinating thing. It is being somewhat oversold for consumer testing right now, but soon we will have the full 3 billion nucleotides available relatively inexpensively.
Wow.
I think for most people who want it. And that's going to drive a tremendous amount of very interesting research, not only medically, but in the genealogy world as well.
That's very interesting.
But we should be waiting, and I support any nonprofit projects that try and use this for research first, and then maybe down the road for effectively hobby reasons.
The problem is getting the money to do the research. Yeah, it is not
Always.
easy.
Well, thank you very much. It's been very interesting, and I'm glad we had a chance to look at the opposite end rather than just hearing one side of it all the time. Thank you very much.
Thank you
Thank you.
very much. That was very helpful.